the tree of life. We're probing one branch of the opisthoconch supergroup which also includes the kingdom Animalia. Okay, so we're down here where it's highlighted in pink. We're in the weeks to come, in the next 2 weeks, we'll cover land plants. For the remainder of the semester, we'll focus on animals and basically everything else, you know, pretty much everything else, including all of the other supergroups we have covered under the protist chapter. So just the closest relatives. So today we'll cover fungi and nucleariids, and then the other groups later. Okay, so they are opisthoconchs, and you can see that we share, fungi and animals share a relatively recent common ancestor, a more recent common ancestor than either of us shares with plants, for example. So with this knowledge, I've got a little, as promised, phylogenetic tree exercise for you all to do. So why don't you take a couple of minutes here and draw a phylogenetic tree. Hopefully you have a pencil and paper in front of you that includes just these 4 clades, okay? Animalia, plantae, fungi, and bacteria. And while you do that, I'm going to go ahead and unlock permission under chapter 24, because I've just been informed that that's locked. Thanks, Julia. So how would you approach this particular exercise? Somebody kind of speak up and tell us what your thought process is. You'd make it rooted, okay? Meaning that you'd start with a single branch at the bottom, right? Like a vertical branch, okay. And then what would you do, Becca? What's a good approach to this? You guys can turn your mics on and pipe up. So one thing in a phylogenetic tree to realize that is that, you know, if there are no known polytomies, right, you know that with every tree you're going to have a basal taxon, and that's the one that broke off first, and you're going to have a pair of taxa that are sister taxa, that are one another's closest relatives, right? So among these 4 clades, and that's all you need to put in the tree. You don't need to put anything else, okay? This is not, you're not making the tree of life. You're making a tree that only includes these 4 taxa. That's all you're considering today in this exercise. Which one is the basal taxon of these 4?
Bacteria.
Bacteria, very good. And why did you pick bacteria, Josie?
Because the other 3 stem from archaea.
because the other three, say that again?
Aren't the other three in the archaea branch?
No, the other three are what?
Oh, eukarya.
Yeah. You
That's
meant
what I
to say eukarya, right?
Yes,
Okay,
yes.
good. Yeah. Oh, very good. Okay, so yeah, basically, when you look at this list, you can, you can break up these 4 taxa into 2 groups. So group 1 is these 3, Animalia, Plantae, and Fungi, which are all eukaryotes, and bacteria, which is a bacterium, right? So if you're going to do, and this is going to be a diagonal tree, basically, because diagonal trees are, I think, the easiest to do, okay, easier than vertical or horizontal trees. I think that's where some of you got in trouble with the way you represented or failed to represent the polytomy that exists at the base of the eukaryotic tree, right? It's just a drawing thing. So it splits, right? There was a split a long time ago right here. Okay? And the split is into 2. If there's no polytomy, if we know what's going on, there should be always a split into 2, not more than that. If you have more than 2, then you've got a polytomy on your hands. That's when you have a polytomy on your hands. Okay, so you know that bacteria is the basal taxon. We're not even talking about archaea. Basically, we don't have to do anything with this branch, but we sort of know, we understand that this branch, that everything from here on up is eukarya. Okay, everything is a eukaryotic organisms, plants, animals, and fungi. Okay, so now we got to finish up that branch, which would be the next one to come along. Out of
Plants.
the plants. Okay, very good. Very good. Plantae, because as we've just established, the sister taxa are animals and fungi. They are one another's closest relatives. Okay, so that means that the next one to come in line is the kingdom Plantae. Okay, and then we've got Animalia. And then we've got fungi. And it really doesn't matter whether you put them, arrange them like this, or whether you switch these 2 around. Okay, remember that these points can pivot. Okay, and everything is hunky-dory because our splits, right? We have this common ancestor here. This is the common ancestor to all 4 of these taxa. And it splits, and the split is 2-way. It's a 2-way split, which is good. It's what we want. Okay, again here, we've got the 2-way split. So basically we've got land plants going this way. And remember, land plants are in the supergroup Archaeplastida. So they are archaeplastids. Essentially, this represents the split between archaeplastids here and opisthokonts here. So again, we're not asked to put that taxon on there, but if we were, we could say all of these are opisthokonts. Okay, so meaning from here on up, what we have is opisthokonts. Okay, in terms of traits that make the difference here. So obviously here the trait is a eukaryotic cell. Okay, what are some traits that distinguish plants from animals? So obviously plants can photosynthesize, animals can't. And so, maybe heterotrophy. Okay. Versus autotrophs here, autotrophy, photosynthesis, however you want to express it. Does that make sense? Okay, so hopefully that's a fairly easy and useful exercise. Okay, so coming back here, you make these trees based on the knowledge from this tree right here. So here's our common eukaryotic ancestor. And when you're looking at a horizontal tree, all of this, the base of the tree, the fact that you have 5 lines, 1, 2, 3, 4, 5 lines departing from it means that you have a giant 5-way polytomy. 1, 2, 3, 4, 5. So this entire vertical line right here is like a single point. Okay, that's what represents your polytomy. And you see that when it comes to the comparison between fungi and animals, there is a more recent common ancestor between those two, right? They're both opisthocons than there is between fungi, animals, and land plants. Okay, land plants are here, and to find the common ancestor to all three groups, you have to go all the way down to the base, to this line, to the base of the eukaryotic tree. Okay, so hopefully that helps a little bit conceptually. Okay, so if I were to characterize a fungus, using biology words, talk about what it is. They are heterotrophs that feed by absorption. That would be a good way to describe them. In many ways, fungi and animals are similar. They're both heterotrophs. But one key difference is that unlike animals, fungi do not ingest and digest. Okay, so animals ingest food and we digest it. Fungi kind of do that, but in the opposite order. What they do is that they produce and secrete hydrolytic enzymes, digestive enzymes, into their surroundings. Okay, the enzymes break down complex molecules, which are then absorbed into the fungus body. Okay, so they just take what they need. In a sense, they don't need a digestive system. They don't need to poop because they only absorb what they need. Okay, so it's the reverse of what animals do in one sense. Fungi can be said to digest first and ingest second, if that makes sense. Okay. They can be decomposers, parasites, or mutualists. So let's review some of the terms on here because there's some heavy duty biology terms here, which by the way, I would love to start seeing in some of your field notebooks, okay? Now that we're into multicellular organisms, I'm sure that you're coming across fungi or fungi symbiosis, fungal and algal symbiosis that we call lichens. And it would be great to see some of that vocabulary in your field notebook entries. So heterotroph, decomposers, parasites, mutualists. So heterotroph refers to how you get your energy and your building blocks. Okay, so this is a term about energy and essentially the carbon source, right? Building blocks. That's what I mean by that. Carbon-based molecules, the molecules of life. So autotroph, it means you get these building blocks from inorganic places, and you get and usually you get your energy from usually the sun. Okay, photoautotrophs in the case of photoautotrophs. Heterotrophs, you get your energy and building blocks from other organic compounds, okay, from other living things, living or formerly living things. These other terms, decomposers, parasites, or mutualists, those are terms from ecology. And so they describe interactions relative to the organism's environment. Okay, so remember that decomposers are the ones that close the loop, right? So when we talk about these building blocks right here, these carbon-based molecules, they cycle through an ecosystem, right? From producers to consumers to decomposers. And it's basically these decomposers that close the loop on getting these elements, these atoms of elements cycled through an ecosystem. So it's kind of a job within the ecosystem. Certainly some of them are consumers, none of them are producers. Some fungi though are consumers because some of them are parasites. So a parasite is a heterotroph. These two terms right here are also ecology terms and usually used to denote a type of symbiosis. So remember, symbiosis is where two species live in very, very close physical association with one another. Whether they're parasites or mutualists describes what the nature of that relationship. So a parasite is a type of symbiotic relationship wherein one species benefits, but the other one is harmed, okay? And that contrasts with mutualism or mutualists whereby both partners in that symbiosis benefit from that association. A little bit about their body structure. It seems kind of weird to talk about fungal bodies, right? What is their cellular form? Some species exist as multicellular filaments, but some species exist as single cells. So anything that we call a yeast is a fungus. Okay, so brewer's yeast or baker's yeast, whatever, that's all. That's a fungus. And in some cases, they exist as both in different parts of their life cycles. Multicellular fungi form usually a really thick, really tight network of filaments called hyphae. So this is a form fits function thing. So remember, form fits function is one of the major themes in biology. It's the idea that you can deduce the function of a particular structure by analyzing its structure, and you can do the opposite. Right? You can look at the function of something and deduce what shape it should have. Okay. So if the way that you obtain your nutrients is by digestion first and absorption second, then absorption is everything about how you get your food. And if you're going to absorb from a surface, it sort of stands to reason that you would need an enormous amount of surface area. Okay, and so hence the hyphae, these tiny, tiny, almost microscopic hair-like structures that consist of basically tubular cells. Okay, so these tubular cells have plasma membranes, but they also have cell walls on the outside of them. So this is another characteristic of fungi in this group, and they are reinforced by a polysaccharide called chitin. So chitin contains a nitrogen appendage. Basically, it's strong, but it's also flexible, and it helps with feeding by absorption. By keeping cells from taking on too much water and potentially bursting because of osmotic pressure. So in that sense, the chitinous cell walls function in a similar way that they do in plant cells. Chitin is also a major component of the exoskeleton of arthropods. So you see a cicada there, and it's just molting. Okay, we'll see that an entire group of animals does that. They molt when they as part of the growth process, and so that little shell that you see behind is almost entirely made of chitin. Okay, so the chitin monomer is what you see there on the right. You recognize that green hexagon, which should remind you of glucose. So it's basically a glucose molecule with in blue that nitrogen containing appendage. Okay, so you put a whole bunch of those together and you get chitin. Okay, so when we look at a multicellular fungus, what does it look like? What does its body as a whole look like? So it turns out that the hyphae, those tiny, almost microscopic filaments that are pictured here, form an interwoven mass called a mycelium. Okay, mycelium. And that mycelium penetrates and infiltrates the material on which the fungus feeds. Okay, so there might be bits of wood in here, there might be, you know, dead things or whatever. And by reaching in between every little nook and cranny, the fungus can take advantage of those nutrients. And so the mycelium is like the body of a multicellular fungus. Okay. And so here's kind of a real life example here. What you see on the right is an oyster mushroom. I was at the grocery store last night and I saw that they were selling these beautiful oyster mushrooms. And what you buy really only represents a really small and really temporary fragment of the organism. Okay, so the main body of this organism, in fact, is the mycelium. It doesn't have tissues, okay? It doesn't have organs the way plants do or the way animals do. Doesn't even really have recognizable parts, right? There are some protists, some multicellular algae that have, they don't have separate tissues, but they have distinguishable body parts, right? They'll have the stipe and the blades and things like that, the holdfast. Here it's all just mycelium, okay? Fungal hyphae microscopically come in two different types, either septate hyphae or cenocytic hyphae. Let me write this down. So septate is what is found in most multicellular species. They basically they have walls that separate the individual cells. So again, septum is the singular. If you're talking about a whole bunch of them, these walls here that separate them, kind of see them, that would be septae. Okay. And the septae themselves have little pores in them or little holes. Okay, so if I draw two adjacent cells like this, let me come in here. Oh dear, my eraser. Here we go. There would be within the septum, there would be these little pores. So that fluid and sometimes organelles can actually pass through. All right, the second type is cenocytic hyphae. So you pronounce it like this, cenocytic, cytic. Hyphae. So that's like one giant cytoplasm with multiple nuclei, which should remind you of something, right? Does that remind you of anything? That body structure with cenocytic hyphae. What does that remind you of? Plasmodial slime molds. Plasmodial slime molds. Multicellular, not really. Single cell, giant single cell in the plasmodial slime mold with multiple nuclei, just like a big blob. Some fungi have specialized hyphae, allows them to do some pretty cool things. Often they have to do with symbiosis. So some mutually beneficial relationships are called mycorrhizae. So mycorrhizal fungi are mutualistic because they provide their host plants with phosphates and other minerals. And in exchange, the plants supply the fungi with carbohydrates, right? Because plants can really only make carbohydrates, right? Photosynthesis involves taking in carbon dioxide and water. Carbon dioxide and water contain the elements carbon, hydrogen, and oxygen. They don't contain nitrogen, they don't contain phosphorus, they don't contain potassium, they don't contain any of the essential elements that are essential for life. And so that's what the fungi provide the plants in these mycorrhizal relationships. And in exchange, they get carbohydrate nutrients um, from their host plants. This should give you pause. Some fungi have highly specialized hyphae that actually enable them to feed on living animals. For instance, when a nematode, which is what you see here, a nematode is a type of very tiny unsegmented worm, slips through these specialized hyphae, these swellings develop that you can see on this scanning electron micrograph, and that traps the animal. At that point, other specialized hyphae begin to develop and penetrate the animal's body and begins to digest it. On the mycorrhizal fungi, we distinguish between a couple of different types. And this will come up again in the chapter on plants. But basically, the two types are illustrated here. So at the top, what you see here are ectomycorrhizal fungi. Let me write that down. Ecto meaning outside, myco, fungus, mycorrhizal. Rhizal, same root word as root, actually. And there's two R's. It's hard. Mycorrhizal fungi. Basically, they form sheaths of hyphae that kind of wrap around the surface of plant roots. But they also those hyphae do grow into the extracellular spaces on the outer part of the root. So that's what you can see in here in blue, is that they squeeze in here past and in between the plant cells. Okay, so you can kind of see that there would be some mechanisms here whereby the plants would share some of their carbohydrates with the fungi, but the fungi would then in turn share some of the minerals and nutrients that they absorb as well. The second type is called arbuscular. So arbuscular mycorrhizal fungi. And so these form these branched structures called arbuscules, okay, kind of like minuscule, okay? This is an arbuscule. Same root word as arbor, okay? They look like tiny little trees, and they poke through the roots, cell walls, and into tubes that they form by pushing in the plasma membrane. We'll see a detail of this again in a little bit, but that's an example of a specialized hypha. Questions? All right, so that's body structure.